Okay, my name is Samantha Capel. I'm from the University of Illinois. And today I'm going to talk to you a little bit about how RAD sequencing might be used to assess and improve species translocations. So in response to rapidly accelerating extinction rates, conservation managers have employed an array of strategies to combat species losses. And among these, translocations, or the artificial movement of individuals between wild populations, have become quite common. And although they're employed quite frequently, success of translocations is often elusive or unclear. And so, in some instances, the success of translocations that have been attempted to be assessed using generally more traditional genetics methods such as microsatellite markers or mitochondrial DNA. But these methods might not give us as deep of insights we might need or want in order to really determine if a translocation was successful or not. And so moving away from traditional genetics methods to more advanced, more recent genomics methods such as RAD sequencing will allow us to more definitively determine the success of translocations. It allows us to look deeper into the genetic effects of translocations on the recipient population, and it may help us maximize the success of translocations by choosing the most appropriate source populations prior to actually implementing these management plans. And so I'm using a natural system that's experienced extensive translocations, the greater Prairie chicken. And so this is a grouse species that prior to European settlement of the Great Plains was virtually ubiquitous throughout the Midwest. But once cropland and rangeland started to dominate the landscape in the late 1800s, ranges quickly contracted down to what we see today. And so of the states that have existing prairie chicken populations today, Illinois suffered some of the greatest losses. They went from an estimated 2 million birds statewide to less than 50 individuals by the early 1990s in 2 small isolated populations. And so in response, the Illinois Department of Natural Resources implemented a series of translocations from 1992 to 1998, and they moved birds from existing populations in Minnesota, Nebraska, and Kansas in the numbers that you can see here. So just on this little demographic trend, we can see in the shaded regions, these are years that we have translocations implemented into these 2 existing populations in Illinois in Jasper County and Marion County. Approximately 10 years after the completion of the translocations, the genetic diversity was assessed in the Illinois population to assess the success of this translocation. And so in all of these graphs, lighter shaded bars simply represent estimates for all of our source populations, while darker shaded bars represent those for our Illinois population prior to and then after the translocation. This little dashed bar is simply just the statistic average for our source populations and might be thought as maybe a goal of what we would want to see in something like a translocation. And so, while in some instances we have a nice signature, a nice biological signature of yes, it seems that maybe genetic diversity did increase, in many other instances we have a really unclear pattern, and this makes it really difficult for us to determine if yes or no was this translocation successful in terms of increasing genetic diversity of this population. And so what I did is I went back to these same exact, some of the same exact samples that were analyzed previously and performed RAD sequencing on them. And so I have over 100 samples from all of these populations that were involved in the translocations. We did a double digest RAD library prep on these. This library was sequenced on 2 lanes of NovaSeq 6000 using paired end reads, and raw reads were fed through the stacks analysis pipeline using the referenced align method. I filtered, just as one of the many filters, we used this threshold of, I'll just say. We used a threshold of at least, a loci had to be present in at least 80% of individuals for any given population to be considered, and this left me with just over 200,000 SNP loci across individuals and populations. And so the first thing that I did is I went back and I recalculated some of these same exact measures that were assessed in previous research. And one thing that we find is increasing our number of loci by orders of magnitude allows us to much more accurately estimate the levels of genetic diversity. And so what I find is just over a 20% increase in our expected heterozygosity of our admixed Illinois population. And this is a very highly significant difference when we compare the pre- and post-translocation populations, whereas in previous work they found less than a 10% increase, and this is a non-significant difference. We also farly increase the precision of these estimates, which we can simply look at by comparing the size of our error bars between these two methods. And so by greatly increasing our precision and our accuracy in estimating measures of genetic diversity, we can much more definitively determine the success of translocation simply in the terms of, was genetic diversity actually increased? We can also look deeper into the population structure of these populations using RAD-seq. And so in previous work, they ran structure and tried to group these populations by genetic identity. And what they really only found was two main genetic clusters of groups, with Illinois grouping away from all of our source populations. I took just a subsample of 4,000 SNPs from my data set, and I find evidence of finer population structure. So once again, we have Illinois clustering away from our source populations, but now we're starting to see finer population structure within our source populations. We can also more precisely track the contribution of each individual source population to our admixed population using private alleles. So just very quickly, if we were to sample and sequence each one of these populations at the same locus, an instance of a private allele would be that where all but one population share the same allele, and our sole population here would have a unique or private allele. And so when we move individuals from a population like Minnesota into Illinois, if we resample the population some years after, what we should find is now the presence of both of these alleles, and we can definitively say that yes, individuals that were translocated from Minnesota did successfully interbreed into this population. And so they did attempt to look at private alleles with previous work, but once again, simply due to the nature of only looking at a handful of markers, they didn't find many instances of private alleles among their source populations. And so not surprisingly, looking at my full data set of 200,000 SNP loci, I find many more instances of private alleles. So then when we look at the Illinois 2003 or the Illinois post-translocation population, in previous work, they were really only able to draw general conclusions about what the relative contribution was of those translocated individuals. Whereas now I can actually look at the Illinois population and count how many of these private alleles do I now find in that Illinois 2003 population. If we normalize these just to a relative percentage of what was previously found, of what these populations contain that were found, then in the Illinois 2003 population, and relate that to the number of birds that were brought from each individual state, we do see somewhat of a general pattern where the state that contributed the fewest number of birds also contributed the smallest percentage of private alleles. The reason that we might have similar results between Kansas and Nebraska here is because they have some overlapping range. There may be some evidence of gene flow between the two, and so they may not be all that genetically unique from one another. So the genome-wide distribution of markers in RAD sequencing also allows us to look at functional differences between populations. And so this is something that's not necessarily always considered seriously in species translocations. The presence or potential presence of significant adaptive variation among these populations is not always considered. And so this is something I'm very interested in, and simply due to the large geographic distribution and the environmental variation that exists among these populations, I find it very unlikely that significant adaptive variation did not exist among these populations prior to translocation. So this is something I'm currently looking into. So what I hope to do is detect and compare loci that show significant evidence of adaptive variation between the source populations and the Illinois population prior to translocations. If I do identify these loci that appear to have significant adaptive differentiation between these two sets of four populations, I'm going to then look at the effects that the translocations then had on this Illinois 2003 population. If Illinois did possess some unique variation, that may have conferred some adaptive advantage, which has now been replaced by source populations, which is what I suspect, this may have negative consequences for our population and may help to explain some of the continued declines that we see in our numbers of these populations following translocations, and even in the presence of increased restored habitat in these areas. And ultimately, if we're admixing these populations to a point where we're replacing a local adaptation with that of others, we may have this misalignment of adaptive alleles in local environment, which will ultimately have negative consequences on our population, which is obviously what we don't want in a translocation. And so just some preliminary data that I have on this point. What we're looking at here is smooth FST values across one of our scaffolds. And so we have 3 different population comparisons on this graph. And so in green, we have differentiation between Illinois pre-translocation and Kansas. In purple, we have Illinois pre-translocation and Illinois post-translocation differentiation. And then finally, we have Illinois post-translocation compared back to that Kansas population. And so in some instances, in some stretches of sequence, we see these low levels of differentiation, which are likely indicative of background levels of differentiation due to neutral processes like something like drift. And in other regions, we see potential targets of differential selection among populations. And so if we look a little closely at this graph, what we see in our green line is that we have significant levels of differentiation between the Illinois pre-translocation population and Kansas. When we look at the 2 Illinois populations prior to and after the translocations, this pattern of differentiation persists. And then when we compare that Illinois post-translocation population back to Kansas, we're now back at these low levels of differentiation, which are similar to our background levels. So what this tells me is that the adaptive, or not necessarily adaptive, but the variation that existed between these populations has now been replaced in Illinois by the Kansas alleles. If this is indeed a target of differential adaptive variation, and that maybe this sequence or parts of code in the sequence have adaptive function and then they lie near or within coding regions, this may have potential negative consequences for our population now. And so what I hope to prove when I'm looking further into evidence of adaptive variation and the potential consequences in the admixed populations is I hope to explore this idea of pre-screening really our populations for something like adaptive variation before we even implement something like translocation. So this would simply mean that we would go to potential source populations in our recipient population and collect genetic samples. We would analyze these samples and look for significant evidence of adaptive variation between the recipient populations and any of these source populations. And this might allow us to then choose our most appropriate source population, effectively trying to balance an overall, this goal of overall increasing our genetic diversity while not mixing populations that show significant evidence of adaptive variation and potentially causing maladaptive consequences in our admixed population. And with that, I would like to thank my advisor and my committee members. I would like to thank people that were extremely generous in donating samples. They're all from the 1990s. Without this, the study would not be happening. I want to thank my funding sources, and with that, I will take questions. Yeah.
I wonder, so I agree completely about the idea that we can pre-screen all populations beforehand. And I wonder what genetic evidence you might need to decide not to do a translocation, and
Right.
what your thoughts are on that.
Right. So some of the environmental variables that I would be concerned with such a wide geographic area, something like differences in precipitation. It's extremely important for a species like this. Some of the things that's most important for juvenile survival is how wet the season is. And so the basic goal is do we find areas in the genome that have this really significant differentiation? Do we find that they may have adaptive significance? So I'm lucky in that I have a reference genome that is annotated. The nice thing about something like STAX is we can do non-referenced based analyses. And so trying to look for maybe an enrichment of areas that show a significant differentiation that share maybe adaptive functions. And if this seems like maybe it would be extremely important in those two different environments, then maybe we would want to stay away from a population like that. Does that make sense?
Thank you.
Thank you very much.